Welcome, everyone, to StabilityHub's webinar on stability testing challenges for N-Nitrosamine impurities. Our presenter is Geoff Carr, pharmaceutical consultant and StabilityHub contributor. Our panelists today include Patrick Kelleher, Associate Director, Physical Sciences at Freethink Technologies, Incorporated, Walter Routh, founder of Sagana Consulting and Associate Editor at StabilityHub. Walter is participating by typing us today with the loss of his voice. And yours truly, John O'Neill, founder of the Pharmaceutical Stability Discussion Group and editor at StabilityHub. A few housekeeping notes. It adds a lot to the event if you can turn on your camera. We can deal with passing children and cats, and it's fine to have your lunch while you're muted. Participants should stay muted unless invited by the moderator to unmute. Questions and comments can be submitted using the chat feature. We may also conduct polls through the Zoom raised hands feature shown on the attendance roster. Just be ready to quickly remove them as soon as the count is taken so they don't throw off a later count on the polling. This meeting is being recorded and will be posted in the articles and video section at stabilityhub.com. The monthly newsletter article of the same title is available at the site as well, if you care to do a more detailed reading on the topic. With that, we'll turn the program over to Geoff and encourage you to submit your questions via chat at any point in the presentation as they pertain to the slide or topic being covered. Take it away, Geoff.
Okay, thanks very much, John, and good morning or good afternoon, ladies and gentlemen. So today we are going to talk about stability testing challenges for N-Nitrosamine impurities, which hit the world about 7 years ago. So N-nitrosamines are potential carcinogenic substances, and so and because of that, they're highly undesirable in any pharmaceutical products, and even at levels way below the regular ICH impurity levels, or impurity thresholds rather. So in about 2018, traces of N-nitrosamine impurities were starting to be observed in some batches of sartan products, and this led to considerable international concern. So many different agencies got very concerned about all this. Soon after, it turned out that these were becoming potential impurities in a number of other pharmaceutical products, and it was developing into a very significant issue for both the industry and the regulatory agencies. In this presentation, I'm going to summarize the issue, plus associated analytical and stability study challenges as follows. We're going to look at an introduction, which is just about done now. What are N-nitrosamines? Some background of the issue. Some more recent developments, which include the classification of nitrosamine impurities and the establishment of acceptable intake levels. I'm going to look at some analytical chemistry challenges and then see if we can conclude anything from all of this. So, moving on to what are N-nitrosamines? Okay. So, nitrosamines are readily formed by nitrisation reactions between amines and nitrite ions. This happens preferably at low pH. So, here's an example of a generic secondary amine, sodium nitrite in the presence of something like HCl. And you get this compound here, which is a nitrosamine derivative of that secondary amine. It's a very standard reaction used by synthetic chemists. And in a synthesis procedure, these reactions do proceed pretty rapidly at ambient temperature. In fact, they may even require cooling because they do go pretty quickly sometimes. And they generally provide good yields of products, which in this case are pharmaceutical products we definitely don't want. Secondary amines are the most susceptible amines for nitrosamine formation, but there have been reported observations with tertiary and even quaternary amines that apparently led to batch recalls. But for this to actually occur, it really would require that you have an initial or concerted dealkylation step within the reaction mechanism. Otherwise, you couldn't form a nitrosamine. So it's not so straightforward to happen. Primary amines are not susceptible to this type of nitrisation reaction. With primary amines, these do react rapidly with nitrites, but the products are diazonium ions, which fairly readily degrade by elimination of nitrogen. With aromatic amines, the diazonium ions are more stable and, in fact, form the basis of the whole dye industry many years ago. Any chemical structure that includes an N-nitrosamine group is going to be considered to have a potential carcinogenic activity. Therefore, reports of these impurities in pharmaceuticals did lead to considerable concern within the industry and regulatory agencies, as carcinogenic impurities get very strictly controlled in pharmaceutical products. The acceptable intake for these things are generally way below what's allowed for non-carcinogenic impurities and are likely to be in the parts per billion type of level. Guidelines have been introduced on this topic, generated by FDA, Health Canada, EMA in Europe, WHO, MHRA in the UK. All of these agencies have produced some sort of guideline, and perhaps others have also. So, some background of the N-nitrosamine issue. So, first detected in sartans that contain a tetrazole ring in the chemical structure, specifically valsartan, where it was found to contain traces of nitrosodimethylamine. This was the first example. This is the structure of valsartan. A tetrazole ring is this 4-nitrogen 5-membered ring. And initially, it was thought that the cause of the nitrosamine came around the tetrazole synthesis step in the preparation of valsartan. So, in order to generate tetrazoles, sodium azide is a commonly used reagent, and in reaction with nitriles produces tetrazoles. So, here's a brief example. This is benzonitrile. It can react with sodium azide, producing this phenyl tetrazole. When you do a reaction like this, it's important to remove any excess reagents following the reaction, and sodium nitrite is commonly used for this purpose. It was suspected that it was the quenching reagent reacting with traces of dimethylamine residues that led to the formation of nitrosodimethylamine in valsartan originally, and that was considered to be the cause of this contamination. Okay, this was a possible explanation, but it then rapidly became apparent that this was not the only example, and it was more complicated than this. So further investigations led to observations of nitrosamine impurities in a lot of other things. There were other sartan products that did not contain tetrazoles in their chemical structures, and not long after, it was also found in things like ranitidine, nizatidine, metformin, and it goes on and on. And we were starting to see a lot of product recalls, which is not a very desirable thing to have happen around here. Initial observations did suggest that it was a problem due to side reactions during drug substance synthesis, but subsequently it was found to be a much broader issue than this. So it was quite clear that in order to get a nitrosamine, we needed a secondary amine, although tertiaries and quaternaries apparently do work also, but they're going to be less efficient. And you also need some nitrites. But these things can come together in various ways. The source of amines during synthesis could be starting materials, intermediates, finished API, synthesis byproducts, API degradation products, some reagents, some solvents. All of these have the potential to contain amines which are nitrosatable. Sources of nitrites, again, they could be trace impurities in synthesis reagents. They could be trace impurities in pharmaceutical excipient ingredients. They could actually be in the reagents themselves. They could be leachables coming out of packaging. Nitrites are all over the place in small ppm quantities, and so they could come from just about anywhere. So nitrosamines could certainly be created during synthesis, there's no doubt about that, but there was also the possibility that it was for them to form more slowly during the product shelf life by various interactions. And because of this, it makes it a topic to be addressed during stability testing. And so of great concern to us stabilitarians and the Stability Hub. Acceptable limits for these compounds are in the parts per billion type level, and I'll put more about that later. It only requires the presence of ppm levels of the amine and nitrite components to generate unacceptable amounts of nitrosamine. So you only need very, very small traces, and way below the ICH limit levels for regular impurities, there would be plenty enough there to get you ppb levels of nitrosamine. So even nitrosation reactions that are extremely unlikely could still be capable of gradually generating unacceptable levels of nitrosamine for products which are being held at ambient temperatures and have shelf lives that could be 2 to 5 years. That's plenty enough time for ppm levels to generate unacceptable levels of nitrosamine. So definitely a level that needs to be addressed during stability testing.
Hey, Geoff, we have a question that seems to go along the lines of what Patrick is doing. Can long-term growth of the N-nitrosamines on stability be predicted, for example, with a tool such as ASAP? And then how acceptable would that be by regulators in lieu of the traditional stability data, which seems like it would take 2 to 5 years, spanning the target shelf life? Yes, Patrick,
Yeah,
that question.
it could be. Yeah, it is a possibility, but because we're dealing with the requirement for the ingredients to be just present in ppm levels, so the amine, the nitrite that's required, only needs to be present in ppm levels to do this gradually. It might be quite difficult to set up the ASAP reaction conditions to properly model this.
Actually, Geoff, I'll add,
But in principle, it probably could be done. Yeah, sorry.
and we do have experience using an ASAP study where we're looking at a nitrosamine. I think predominantly what we've been looking at, and I think you're going to get into this later, is the drug substance related nitrosamines, the NDSRIs, rather than the small molecule ones. But yeah, we have experience doing a number of ASAP studies. There are analytical challenges, which I think you'll also talk about. But as long as we're able to generate sufficient data, we have been able to accelerate formation of nitrosamines in a predictable fashion, use moisture modified Arrhenius equation to predict rates back at long-term storage conditions in packaging. This has been quite successful. There's a few added considerations when dealing with nitrosamines. One of these becomes, as you described, you need a nitrite present. We have seen instances where it appears that the nitrite is a limiting reagent, so you get some growth of a nitrosamine and then it stops, it levels off. We believe this to be because you've consumed all the available reactant and now you stop. But we have, for a number of our customers, run ASAP studies on drug product, measured nitrosamines using LCMS, and built models. Anecdotally, we have at least one case where we've supported a customer who had a late stage product going commercial where they discovered a nitrosamine very late in the game. very scary thing for a developer. And they were able to reformulate their drug product by adding in a small amount of antioxidant. We did ASAP on both the new and old formulations, showed the antioxidant successfully mitigated the nitrosamine risk. And we're able to, using their long-term data on their original formulation and very limited data on their formulation with antioxidant, go back to regulators and get approval. So regulators have seen this type of approach. They have accepted it. There's a couple publications out there by pharma working groups and the FDA describing acceptance of accelerated stability modeling for nitrosamines, be it ASAP or any other variant thereof. So
thank you,
it is applicable.
Mm-hmm. Much appreciated.
Okay, thanks, Patrick. I need to move on. Okay, so in order to cope with this, the FDA introduced what they called a three-step mitigation strategy. The FDA introduced it, but other agencies are also doing the same sort of thing in other territories. And so this is quite now a widespread strategy that's going on. And what's required is that it's applied to all companies' marketed products. So it's a huge burden on all companies to deal with this. But basically, the three-step mitigation strategy is: one, they have to do risk assessments on all products, and this is essentially a paper exercise. Don't have time to go into great detail on this, but what you have to do is to go through every aspect of the synthesis of your API, and then the manufacture of your product, all the ingredients that have been used, the background of all those, and make an assessment on what is the risk that there could be some secondary amine or some amine there which is nitrosatable, and what is the risk that there could be sufficient nitrite present which would do that nitrosation for you. When you do this, stage one, which is quite a lengthy process, you then have a decision: is there a risk or is there no significant risk? If there's no significant risk, no further action is required. If there is a significant risk, you are required to then deal with confirmatory testing of your products to go looking for possible traces of nitrosamine. And again, if none are found, you can assume that therefore everything is okay. If some is found, then you are required to deal with it by redevelopment of the steps that are causing it, possibly variations on your product license, process variations would be required, and then it becomes a registration issue to file for process variations. It's a huge exercise for all pharmaceutical companies, and I assume it also makes quite a big job for the regulatory agencies. So you might think you are now getting the hang of the problem, and I'm going to spoil that comfort feeling.
Hey, Geoff, just before you move on, there's a question on our chat. Okay. What inspection level from the agency can we expect for phase one? Will they critically evaluate the decisions, or will they just be making sure that you did it, or somewhere in between? So when you said that the governments are interested, do we know whether or not they're doing a detailed inspection of those exercises that we do?
Okay. My take on that is that you are not required to send all of your risk assessments to the regulatory agencies. They would be overwhelmed by bombardments of paper to go through. You are not required to do that. Deadlines were set by different agencies as to when it had to be done, but you just have to make sure you get it done and you have the results of it on file. What then might well happen is that when you get an inspection from the agencies, they may well want to look through the assessments that you've done to make sure that you did a decent job of it. But as I said, my take is you are not required to send all of these to the regulatory agencies. But if anybody out there has different experiences with this, I'd be very interested to know about it.
Thanks. Looks like you're about to deliver some bad news to us on this next slide.
Yes, I am. Pharmaceutical, well, this is probably quite well-known news that pharmaceutical science issues are rarely simple. They are usually quite complicated things to deal with. And in this is not an exception. So I've just been emphasizing over and over again that in order to get nitrosamines, you need to have amines and nitrites present to get the reaction. The bad news is that it is possible to get nitrosamines in the complete absence of any nitrite ions, but it does require special circumstances. And this has been recognized by the FDA. I seem to recall seeing somewhere in their latest guideline, they actually do make a mention of this. It does require special circumstances. So your drug substance would need to contain a hydrazine group in its chemical structure. Now, you don't see that very often. Hydrazine groups are very unusual features in pharmaceuticals, probably because they're also pretty carcinogenic things quite often. However, it can happen. There are a few out there, and one example is the anti-Parkinson's drug carbidopa. Okay, this is carbidopa. And this bit here, the two nitrogens joined together is a hydrazine group. This sequence here forms a nitrosamine. So the hydrazine can be oxidized, no nitrites required, but oxidation of a hydrazine could lead to this. It could also lead to other things as well, but this could be a possible product of a hydrazine oxidation, which is a nitrosamine of this secondary amine, and so would not be a desirable thing. Very rare to happen, but you need to be aware that although the guidelines tell you to do certain things, you should also think about the spirit behind what they're trying to achieve. And maybe there are things that could happen that they have not addressed and you should also be dealing with. Early regulatory guidelines tended to concentrate on a small group of specific nitrosamines. Nitrosodimethylamine, diethylamine, methylphenylamine. The list is diisopropylamine, isopropylethylamine, dibutylamine, N-methylaminobutyric acid. You'll find that within the guidelines, the FDA, Health Canada, EMA, WHO, they all have the same list of 7 possible compounds in there. Although in recent revisions of the FDA and Health Canada guidelines, they did introduce a new classification, which I'm going to come to a bit later. But these 7 compounds here were considered to be the main 7 things that are likely to cause nitrosamine problems. Which now takes me nicely into dealing with some more recent developments on the topic and the classification of N-nitrosamine impurities. So this new classification was introduced by the FDA and Health Canada in their 2024 revisions of their guidelines. And they included 2 types of nitrosamine. There was the small molecule nitrosamine and then the nitrosamine drug substance related impurity. And let's just take a look at these. The small molecule nitrosamine does include the 7 molecules that I listed on the previous slide, plus any similar types of nitrosamine molecule, but they are not related to the drug substance. The majority of cases of nitrosamine observations that led to product recalls involved nitrosamines in this particular category. And the reason for that is that the parent amines of those nitrosamines do include secondary or tertiary amines that may be used as things like reagents and solvents in drug synthesis. And so trace residues of these batches would be susceptible to nitrosation by equally traced nitrite residues in either the drug substance or the drug product, or in drug product ingredients, sorry. These are those 7 compounds: nitrosodiethylamine, NDMA, NDEA, NMPA, NDIPA, NIPEA, NDBA, NMBA. Now, the NMBA, this nitrosamine-methylaminobutyric acid, might seem out of place here. It does not look like it's a straightforward nitrosated derivative of anything that's commonly used, but it is actually has been observed in batches that were subsequently recalled. And the reason for this is that it comes from N-methylpyrrolidone. N-methylpyrrolidone is a pretty commonly used solvent in synthesis. It is a lactam, it is a gamma-lactam, and a gamma-lactam is susceptible to hydrolysis. If you hydrolyze it, you get N-methylaminobutyric acid, which means you've got this secondary amine group at this end of the molecule, and that is nitrosatable to give you N-nitrosamine-methylaminobutyric acid. So that's where this one comes from, and it has been detected in products which were subsequently recalled. The other category is nitrosamine drug substance-related impurities. Now, amino groups are structural features that you're going to see in lots and lots of different drug substances. Many of them even have multiple amino groups in their chemical structures, and all of these could be susceptible to nitrosation by traces of nitrite ions. If that's not enough, we additionally have huge numbers of amide groups which are present in the chemical structures, and amide groups are often susceptible to hydrolysis, creating more amino groups which are also susceptible to nitrosation. So the number of products that you could get nitrosamine derived from these drug substance related, or these NDSRIs, is huge. I just previously, on the previous slide, indicated that the most frequently reported observations of nitrosamine impurities were the small molecule category. In reality, the ones that potentially should be present are really the NDSRIs. There must be far more of these present in products than there are of the small molecule ones, but they are not often reported. And the reason for this is probably that they've so far remained undetected, or not many investigators are really looking for them, because it's pretty difficult to really pick on these and look for them and find them. But I would say, in my opinion, there are probably far more of these types of impurities present than there are of the small molecule variety. There is one early example of an NDSRI that was found in valsartan tablets. Now, remember, valsartan was the product that started this whole issue. So this is the chemical structure of valsartan. You see it has this amide group here. This is susceptible to hydrolysis. It hydrolyzes to give you this secondary amine. This is susceptible to nitrosation, and this gives you this N-nitrosamine, which is a possible impurity in valsartan. It wasn't the one that caused the issues in the first place. That was nitrosodimethylamine, but it has been reported as present in valsartan, which I'll show you in a moment. Now, most of the regulatory guidelines on nitrosamines do recognize this as a source, but no other agencies take it particularly seriously. Only FDA and Health Canada have really gone into great details about these NDSRIs. And both of these agencies are now providing tables of known and hypothetical NDSRIs in a whole range of different APIs that are associated with their 2024 guideline revisions. Now, the contents of the lists by FDA and Health Canada are not the same. So it is actually worth checking out the listings from both agencies. And you can get this, it's not written into the actual guidelines, but it's associated with it. And I'm giving a link here to the FDA listing. And the second link here will take you to the Health Canada listing. And they look a bit like this. This is, the FDA listing is exportable into an Excel file. And this slide is taken from the Excel file where it lists a number of APIs and the nitrosamine that has been associated with it, either actually or hypothetically. Now, most of these are direct nitrosamine derivatives of the API, but you will see a few which are actually nitrosamines of degradation products. So this desmethyl almatriptan, for example, is nitrosated. You will see in the listing, there is also a column about potency category, which could be anything from 1 to 5, which I'm going to go into a little bit later. And associated with the category is a recommended acceptable intake limit or an AI limit, which I'll also go into a little bit later. So the Health Canada, the FDA table contains about, I think it's about 250 entries. The Health Canada table has about 160 entries, but they are not all the same as each other. So it's, as I say, it's worth looking at both of them. And a screenshot from the Health Canada one looks a bit like this. Same sort of list. Here's the API. These are the nitrosamines associated with them. On this particular page, most of them look like they are actually nitrosamine derivatives of impurities. Chemical structures are provided. They are also provided in the FDA listing, but you have to look at the web page listing of the Excel file. And then if you click on the impurity, it will take you to a chemical structure. You also have the same categorization and the acceptable intake limits. So these are very helpful tables and certainly worth following up on. This is another page from the table, and you'll recall a few slides ago I was talking about this valsartan impurity. Well, this is the impurity I was showing you before, which is listed in the Health Canada table. It's not featured in the FDA table. Although I should say, but both of these agencies are keeping their tables updated. So I think the issues I've got here, these tables were dated from November 2024, so they might have revised them since then. And by the way, there's also another valsartan impurity here, which I don't fully understand. It's got a benzonitrile group here, so it looks like it's a precursor to the tetrasol part of the synthesis, but it's also got the the butyric acid side chain missing from it. So I'm not sure why that would still be listed, but anyway, it is listed in the Health Canada table. The other more recent developments involve the classification of the establishment of acceptable intake levels. And this could be established on a case-by-case basis by conducting toxicity testing, which would involve animal testing and probably Ames testing, which is bacterial reverse mutation testing. This is not going to be very popular, acceptable to the industry. It would have to be done on lots and lots of different possible nitrosamine. Each impurity compound would need to be synthesized. The testing would be lengthy. It would add significant delays to NDA filing, and it would be incredibly expensive. So it would certainly be a way forward, but it's not something that anybody's really going to do. An alternative option is to use predictive testing. And both the FDA and Health Canada have included columns which include carcinogenic potency category and recommended acceptable intake levels on rails. And this approach could also be used for unlisted potential impurities. So they don't have to be listed in their tables for you to be able to use this, as you'll see shortly. The potency categorization is a predictive procedure based on structure-activity relationships, or SAR knowledge. So it's been determined that the carcinogenic properties of nitrosamine are dependent on the number and nature of proton distributions on carbon atoms located alpha to the nitroso group. That's one feature. The other feature is the presence of activating or deactivating groups within the chemical structures. Now, both of these feature a scoring system, which leads to a final result of between 1 and 5. Maximum carcinogenic activity is a result of 1. Minimum activity is a result of 5. This is a table which shows the rail values, and for category 1, the recommended acceptable daily intake is 26.5 nanograms, according to the FDA. Health Canada want it to be 18 nanograms, which is actually out of step with what others do. Most agencies seem to settle for this 26.5 nanogram figure. If you have a category 2, it's 100. If 3 is 400, 4 and 5 are 1500. Not sure why there's a difference between 4 and 5. Maybe somebody out there does understand that. I've not found that in the table, what the difference is. But both of them are 1500 nanograms per day. So for Cat 1, the Health Canada has adopted a stricter value for some reason. This means, so if you are dealing with a drug product for which the maximum daily intake is 1 gram, this would equate to 26.5 parts per billion as the maximum level of that impurity. Except for Canada, it would have to be 18 parts per billion. And that's how you would use these tables to calculate your limits of any nitrosamine impurities. The rail value, which is being used for category 4 and 5 of 1500 nanograms per day, is still a very strict limit, but it's consistent with the ICH guideline M7, which is about carcinogenic impurities, and provides 1.5 micrograms per day as a threshold of toxicological concern for any unstudied chemical that poses a negligible risk of carcinogenicity. So just having a nitroso group present is a negligible risk of carcinogenicity. And that would mean that you have to limit it. And the limits are incredibly tight. So how do these things work? The number and nature of proton distributions on carbon atoms are alpha to the nitroso group works like this. So this is dipropylamine, nitrosodipropylamine. This is the amine nitrogen group. This and this are the carbon atoms, which are alpha to the nitrogen group. This and this are the beta carbon atoms. So it's this and this which are the alpha carbon atoms, which are of interest with the number of the proton distribution. And both the FDA and Health Canada agencies provide the same examples in their guidelines of how this factor is applied. So here's an example where we have this phenylmethylamine, or that nitrosophenylmethylamine, and this are the alpha carbons. So the carbon on the benzene ring has 0 protons associated with it. The methyl group has 3 protons. And what you're supposed to do is report it as lowest first as 0,3. And it's with 0,3, it has an alpha hydrogen score of 2. This is an example where we have methyl ethyl nitrosamine, nitrosoethylmethylamine. On the methyl side, on this side, we have 1 proton. On this side, we have 2 protons. So it's reported as 1,2, and that gives you a score of 3. Now, it does not give you every example. I'll come to you in a moment as where you can work with any possibility. The guidelines just give you these as examples. The other feature is the presence of activating or deactivating features within the chemical structures. And this could include for deactivating features includes the presence of carboxylic acid groups, pyrrolidine, a nitroso group in a pyrrolidine ring, a nitroso group in a 6-member ring that includes a sulfur atom, and there's lots of other examples. And the presence of these features leads to a potency category value of +1 to +3. Activating features include aryl groups alpha to the carbon atom, a methyl group beta to the carbon atom. These are the only examples given of activating groups, and they lead to a -1 potency value. Having got the alpha hydrogen score, the deactivation score, you add them together, and what you get is the overall score that you use to decide on the category. There is a commercial software that you can use to do this for you. It's built into the Derek Nexus software, which is provided by LASA Limited. Now, for those of you who might be toxicologists or you have toxicology groups, Derek Nexus is a software product which has been used for decades now to predict the toxicology of different types of molecules. It is issued by, provided by LASA Limited, and they have now included a nitrosamine calculation, and the scoring system that I was just going through before with examples is built in. It was created by LASA and it is built into their software. So all you have to do is to feed the software the chemical structure, and it will tell you what category it goes under. You can get this from the website listed here, lasalimited.org. Okay, so now moving on to the analytical chemistry challenges of all this. So the regulatory guidelines, particularly the FDA and Health Canada, are now providing us with pretty effective tools which will enable decisions on whether nitrosamine impurities whether or not they do exceed acceptable levels, but we still have to have problems here. How do we decide whether our product does contain any detectable nitrosamine impurities? And if yes, what are the chemical structure of those nitrosamines so that we can apply these tools to establish appropriate specification limits? These are analytical chemistry challenges, and they need to be probably addressed using both freshly manufactured batches to establish whether they're coming from synthesis, so as part of release testing maybe, but also the possibility of development during product shelf life, and so stability of testing of stability samples. But how are we supposed to do all this? So when we're dealing with regular unknown related impurities that are not suspected carcinogens, we can then depend on ICH guideline Q3A for drug substances, Q3A-R2 for drug substances, and Q3B-R2 for drug products. And generally, we're working in the ballpark of about 0.2% as limits, specification limits, for dealing with unknown related impurities that are not suspected carcinogens. When you're dealing with 0.2% type levels, generally HPLC procedures with UD detection work pretty well. Now that we've got to deal with these nitrosamine impurities, we're required to operate in a range which could be as low as 26.5 parts per billion or 18 parts per billion for Health Canada. This is now equivalent to 0.0000265%, which is way, way below the capabilities of many HPLC procedures. Even if your nitrosamine is rated as category of 4 or 5, the limit is going to be 1,500 parts per billion, and that's equivalent to 0.00015%. So you're still way below the sort of level that HPLC can work for you. You're going to need to use something else. You're going to probably need liquid chromatography with mass spectroscopy detection, LCMS, to deal with this challenge. Now, fortunately, this is a technology which is now quite widely available in modern analytical labs, but it's not straightforward. It's great for operating at high sensitivities and when you know what you're looking for, but when you're dealing with unknowns, it does get to be pretty complicated. So in order to operate with samples and unknown impurities in samples, it's recommended to operate the detector in total ion current mode. You have to do that because you don't know what molecular weight you're going to be working with, so you have to use TIC mode. This will then record all components present, including the target of search, which is those nitrosamine impurities, for which you're wanting parts per billion limits, but it's also going to be detecting non-nitrosamine impurities, which are likely to have specification limits about 0.2%. So you could be generating really complicated chromatograms with huge number of peaks, although you will have the ion mass information available for each of those peaks. You would then have to do a reanalysis of all those peaks, now using specific ion monitoring or SIM mode, to collect spectra for all the impurity peaks observed in the TIC chromatogram, and then apply fragmentation patterns may be needed to acquire some sort of chemical structure information. What we really want to know is which, if any, of those peaks may contain a nitrosamine compound. And a possibility here might look for any parent ion that generates a fragment due to a loss of 30 mass units. 30 mass units would be equivalent to an N double bond O, which would signal a nitrosamine, or it could signal a nitrosamine. It could be other things as well, but it could very well signal nitrosamine. Well, unfortunately, it could also signal two methyl groups because there are 15 each. But what you're going to need is to have some really good organic chemists and mass spectroscopists available within your companies that can do this job for you. But even then, it's going to be a huge task to go through all of these chromatograms and analyze them in this type of detail. Also, for any specific drug substance or drug product, the potential nitrosamine impurity, could it be a direct nitrosation product of the API, which we saw plenty of those listed in the FDA listing and also in the Health Canada listing, but it could also be nitrosated degradation products or other impurities. So it could get to be a very complicated procedure to follow. Now, fortunately, there are also software systems that might help with that. There is a software system also issued by Larsa Limited called Zenith, which can be helpful in establishing likely degradation products from any pharmaceutical API. And you could look for any degradation product that contains an amino group, and that could be a potential source of a nitrosamine. Expert systems can then predict these degradation products of organic molecules. Zenith is a software that does that. Available also from larsalimited.org. Can't go into details on that now, we won't have enough time for that. But to do all this, you're going to need to have analytical groups who are probably specialists at this type of things. They need to be good organic chemists, preferably with very good mass spec training. And that's what I would propose to set up as a group that deals with these sorts of problems for you. But that just leaves me to my conclusions. So recent discovery of nitrosamine impurities in pharmaceutical products have led to a number of batch recalls, which is a very concerning thing to have happen. Quite rightly, regulatory agencies have to deal with this very seriously and have this class of impurity, carcinogenic impurities, have to be very strictly controlled. The term nitrosamine could cover huge numbers of different impurity molecules, and there are vast numbers of actives that contain secondary or higher order amines that have the potential to form these compounds. They also include amide groups that also have the capacity to generate amines and then nitrosamines. Many of the agencies have attempted to explain the issues and propose remedies, but whatever happens, there are going to be very big burdens to pharmaceutical companies. But if these guidances are not followed well, we are likely to see some batch recalls. So, the current guidelines, particularly FDA and Health Canada, are pretty good, provide very good tools that can be used for conducting this type of analysis of your products, but are going to be very challenging to do within the analytical labs. You need trained chemists, and it's going to still be a very big task for them to go through this. And I would finally just say it could provide good specialist opportunities for contract labs. And I think that would be the last slide. So I would just like to finally thank StabilityHub for inviting me to give this presentation, for everybody, ladies and gentlemen, participating, for your kind attention. Thank you very much.
Thank you, Geoff. A lot of information that's great for us to be delving into. We have a question and goes this way. In general, stabilitarians don't have hands-on interaction with discovering, measuring, evaluating, and preventing nitrosamine-related problems. What should their role be in this arena? And I was thinking, from my part, that stabilitarians by nature have an overall responsibility for the integrity of their products, being the final gateway to product registration. So in that regard, they can be educators, monitors, encouragers, reporters, and interpreters within their organizations, more or less the cheerleaders of the process. Because as you pointed out, there's a lot of details here that many of us are just seeing for the first time. And lots of considerations to take in, especially that list you gave us with regard to the APIs and the in-process agents that are used. A lot of these pop up in manufacturing and others pop up in stability. So I think as stabilitarians, we can sort of bring the question to our company, make sure that all these aspects are getting covered. Do you have any other thoughts to add to that?
Yeah, I absolutely agree with you there, John. And just interestingly, it was just a couple of days ago, I saw in LinkedIn there was a company, I won't mention names, I actually can't even remember the name. There was a company that was advertising for an analytical chemist within their group, and they were intending to do was to actually set up a specialist group for dealing with nitrosamines. So they wanted to set up a specialist group that would be responsible for the development and validation of the test methods, but also for the testing, for release testing, and for doing the stability testing part of it, because it really is a specialist activity.
Okay. I have time for another here. What conditions most commonly promote nitrosamine formation over time on stability? So beforehand, once you finally make it to the stability process, is it temperature, humidity, or the combination of the two?
Well, I think it's a combination of the two that would certainly be required for it to happen. Although in synthesis, you do generate nitroso groups using cooling, so it can be a very fast reaction. The thing that makes it a stability issue is when the two components of the reaction need to find each other. So if the API contains the amine and somewhere in an ingredient or a leachable is the nitrite is hiding somewhere, the thing that really it might control the kinetics of it is the rate at which the nitrite works its way into the formulation, and the amine and the nitrite actually come to meet each other.
Okay, thank you. Let's see, time for one more. What's the future of this look like? It almost seems like it's a fad that people are looking at this in great intensity now. Do you think that'll level off at some point and become less of a feature, or will it continue to grow?
Don't know. What I have noticed with this is that the regulatory response to it has been faster, and much more has been going on than I've seen on any other issue that's turned up. So guidelines are appearing all over the place. There's much more activity on this one than I've seen before. So whether it will just settle down, it's hard to say. Maybe it could do. One thing I would say is that it's only new in that it's only just been that it just got reported in 2018. I reckon it's been around for decades. It must have been around forever because nitrites have been present forever. Amines have been a feature of APIs since forever. So these nitrosamines at parts per billion level have been there. It's only with the increasing widespread use of LCMS or GCMS that we're now starting to detect them. And so it's hard to say where it will go.
Okay, well, thank you. We're just about the end of our time. I wanted to wrap this program up by inviting everyone to join us again on Thursday, September 25th at 12 p.m. Eastern for an exploration of the new ICH-Q1 Consolidated Stability Guidance. And also mention that our FreeThink Technology friends are conducting their 2025 Science of Stability Bio Conference, the 6th through 7th of October in Research Triangle Park, North Carolina. You can get more information from them at stabilityconference.com/bio. And I would ask that you watch for further details on our schedule at the StabilityHub website. And thank you all for attending, for Geoff Carr and his presentation, for Patrick Kelleher and Walter Routh, supporting our answers and our conference. And you are a great audience for your attendance today. We hope to see you again soon. Goodbye, everyone.